How sex changed everything, the secret revolution of complex life. Part 1. The redox world of the mid-Proterozoic. Introduction. A planet in suspension.
Between two cataclysms, the Great Oxidation of 2.4 billion years ago and the Cryogenian glaciations around 720 million, stretches a long, still epoch. Continents weathered slowly beneath a muted sun. Oceans lay stratified beneath sluggish atmospheres. It has been called the boring billion, yet within this quietness life achieved one of its most extraordinary transformations. Bacteria and archaea, the only forms of life for 2 billion years, joined in a partnership that created the first complex cells, the eukaryotes. Within their membranes lay the seeds of all later animals, plants, and fungi. Between about 1.8 and 0.8 billion years ago, Earth entered a long period of stability in both climate and oxygen levels. The atmosphere contained only a small fraction of today's oxygen, enough for simple, oxygen-breathing microbes to survive, but not enough to support large or complex animals. In the oceans, the surface waters held some oxygen, but the deeper layers remained dark and rich in dissolved iron, with little or no oxygen at all. This chemical layering created both challenges and opportunities for life, driving the evolution of new ways for organisms to harness energy and adapt to their changing world.
The redox world.
The Proterozoic oceans were unlike today's blue expanses. Beneath a thin oxygenated skin lay vast green depths loaded with dissolved iron. Sunlight penetrated only the upper few tens of meters. Below, sulfate-reducing bacteria thrived in dark, sulfur-rich waters. Periodic mixing brought trace metals and nutrients upward, feeding photosynthetic plankton. These sluggish chemical cycles formed the crucible in which complex cells evolved. Chemical clues locked in ancient rocks helped scientists understand what Earth's environment was like during this time. After about 2 billion years ago, certain metals such as molybdenum and uranium became more concentrated in ocean sediments, showing that parts of the oceans were starting to gain oxygen. Other elements, like chromium and cerium, reveal that oxygen levels still rose and fell over time. Meanwhile, zinc, a metal vital for building DNA and proteins, became about 3 times more abundant between 2.1 and 1.5 billion years ago, matching the appearance of the first complex, eukaryotic cells in the fossil record. These metals were crucial for key biological processes such as breathing and cell division, showing how changes in the chemistry of the oceans helped pave the way for more complex life. Around 1.8 billion years ago, the chemistry of Earth's oceans began to shift. Waters that had once been rich in dissolved iron became increasingly filled with sulfur compounds, creating what scientists call sulfide-rich or euxinic conditions. This change had major consequences for life. Sulfide tends to lock away important metals like molybdenum and copper, which living things need for vital processes such as breathing and capturing nitrogen from the environment. With these metals in short supply, life entered a kind of nutrient drought, a long period when evolution slowed dramatically. This scarcity may help explain why, for nearly 1 billion years, the pace of biological innovation appeared to stall, a time scientists refer to as the boring billion.
The archaeal foundation.
Modern molecular phylogenies reveal that eukaryotes did not arise from typical bacteria but from within the archaea, a domain of heat-tolerant microbes inhabiting vents and sediments. Somewhere in the mid-Proterozoic ocean floor, an archaeal host cell formed intimate contact with a bacterium capable of using oxygen. What began as ingestion or parasitism became symbiosis. The bacterium persisted inside the host, gradually transforming into an organelle, the first mitochondrion. The host lineage belonged to the Asgard archaea, discovered in 2015 from deep-sea sediments. Their genomes encode actin-like and tubulin-like proteins, small GT parses, and ESCRT complexes, molecular machinery once thought unique to eukaryotes. These genes provide the missing evolutionary bridge between simple prokaryotes and nucleated cells. Rising oxygen levels around 1.9 billion years ago created the right conditions for one of life's most important breakthroughs, the merger that gave rise to complex cells. Geological evidence, including the appearance of red-colored rocks and changes in sulfur isotopes, shows that oxygen was spreading across the planet at this time. In shallow coastal waters, small pockets of oxygen, or oxygen oases, formed near the edges of continents. Here, oxygen-using bacteria and oxygen-hating archaea could live side by side, coming into close contact again and again. Eventually, one of these encounters led to a lasting partnership, endosymbiosis, in which a bacterial cell took up residence inside an archaeal host. This relationship could only succeed once there was enough oxygen in the environment for the bacterial partner to provide real energy benefits, setting the stage for the evolution of complex life.
The oldest eukaryotic fossils.
The fossil record of this transformation is sparse but tantalizing. Spherical, ornamented microfossils with complex wall structures appear in shales of the Grapania spiralis assemblage at 1.87 billion years. Thread-like coils up to 2 mm long, the first truly macroscopic organisms. Later forms such as Tapania plana, 1.63 billion years, show branching extensions suggesting cytoskeletal organization and possible colony formation. Tiny fossils called acritarchs provide some of the earliest evidence for complex, eukaryotic life. These microscopic spheres, ranging from 20 to 300 micrometers across, have tough outer walls and internal structures that closely resemble those of modern algae rather than bacteria. Under powerful electron microscopes, their walls show a distinctive double-layer design made of durable organic compounds found only in eukaryotes. Chemical signatures within these fossils also point to photosynthesis, the process of using sunlight to make food. Because acritarchs have been discovered on several continents, scientists know that by about 1.6 billion years ago, these early complex cells had already spread across the globe. Chemical traces found in ancient rocks add powerful support to the fossil evidence for early complex life. In rocks from the Barney Creek Formation in Australia, dating to about 1.64 billion years ago, scientists have discovered molecules called steranes. These are the preserved remains of sterols, fats that help strengthen the cell membranes of eukaryotes, the same type of cells that make up plants, animals, and fungi today. Importantly, making sterols requires oxygen, so their presence not only confirms that eukaryotic life had evolved by this time, but also shows that oxygen was already present in the surface waters where these organisms lived.
Transition summary.
By 1.6 billion years ago, the basic blueprint of the eukaryotic cell was established: a nucleus protecting DNA, a cytoskeleton for shape and motion, and mitochondria generating internal power. The merger of archaean and bacterium had produced an organism capable of complexity far beyond its ancestors. The stage was set for further symbioses, and for the invention of sex and multicellularity.
Part 2: Energy, Symbiosis, and the Birth of Organelles. The energy barrier.
Life's potential for complexity is limited by energy. Bacteria and archaea, though immensely diverse, face a physical constraint. They generate ATP only across their outer membranes. The surface area of that membrane scales far more slowly than the cell's internal volume, and beyond a certain size diffusion can no longer deliver enough energy. For nearly 2 billion years, this law kept cells microscopic. The evolution of eukaryotes broke it. Energy models show that simple cells, like bacteria, face a hard limit on how large and complex they can become. Once a cell grows beyond a certain size, it no longer has enough surface area to produce the energy needed to run its internal machinery or maintain a large genome. The invention of mitochondria changed everything. These tiny, energy-producing compartments acted like thousands of miniature power stations inside a single cell. A typical eukaryotic cell could contain thousands of mitochondria, each generating its own supply of energy. This massive boost, up to 20,000 times more energy-producing surface area than before, allowed cells to expand their DNA, build complex internal structures like the cytoskeleton, and develop the sophisticated systems of communication and control that made multicellular life possible. Around 1.9 billion years ago, the rise of oxygen gave life access to a far more powerful source of energy. Organisms that learned to use oxygen in their metabolism could extract roughly 15 times more energy from the same amount of food than those relying on older, oxygen-free processes. Even though oxygen made up only a tiny fraction of today's levels, it was enough to revolutionize the way cells produced energy, a change that transformed life's entire biological economy and paved the way for greater complexity.
Endosymbiosis, the mitochondrial union.
At some point, an archaeal host engulfed, but did not digest, an alpha proteobacterial partner. The bacterium brought the machinery for oxidative phosphorylation. The host supplied protection and nutrients. Over generations, genes migrated from the symbiont's genome to the host's nucleus, tying their fates together. The symbiont shrank into an organelle, the mitochondrion. Genetic studies show that modern mitochondria, the tiny powerhouses inside our cells, still carry a handful of their own genes, usually between 37 and 70, compared with the roughly 2,000 genes their free-living bacterial ancestors once had. Over time, most of those genes were transferred to the cell's main nucleus, allowing the host cell to take direct control of energy production and coordinate its metabolism more efficiently. Despite the wide variety of species that exist today, all mitochondria share more than 50 essential proteins, pointing to a single common origin from an ancient symbiotic event about 1.85 billion years ago, when one cell permanently merged with another. The host archaean probably lived in anoxic sediments at the oxic-anoxic interface. In such gradients, hydrogen-producing bacteria and hydrogen-consuming archaea already formed syntrophic consortia. Endosymbiosis may have begun as a permanent hydrogen transfer partnership, the hydrogen hypothesis, stabilized by gene exchange and eventual internalization. Chemical clues from rocks about 1.64 billion years old reveal clear signs of a major biological transition. In these ancient shales, scientists have found molecules such as steranes, squalenes, and hopanoids, chemical fossils that together indicate thriving communities where bacteria produced organic matter and early eukaryotes consumed it. At the same time, changes in the chemical makeup of iron within nearby sediments show that more of it had reacted with oxygen, a pattern consistent with the spread of aerobic respiration, the process of using oxygen to release energy from food. Together, these signals show that oxygen was reshaping both Earth's chemistry and its living ecosystems.
The birth of chloroplasts.
Hundreds of millions of years after mitochondria, a second endosymbiosis occurred. A heterotrophic eukaryote engulfed a cyanobacterium capable of oxygenic photosynthesis. Instead of digesting it, the host retained it as a living light-harvesting organelle, the chloroplast. This event founded the lineage of algae and, later, plants. The DNA inside chloroplasts, the structures in plant and algal cells that carry out photosynthesis, is strikingly similar to that of modern cyanobacteria, particularly filamentous types resembling nostoc. Genetic comparisons show that only about 5 to 10% of chloroplast genes still match those in living cyanobacteria. The rest were gradually transferred to the host cell's nucleus over time. The oldest known photosynthetic eukaryotes, the red and green algae, had already split into separate groups by about 1.6 billion years ago, meaning the original partnership, when a cyanobacterium became permanently trapped inside another cell to form the first chloroplast, must have happened slightly earlier. Chemical traces found in rocks from the 1.1 billion-year-old Xiamling Formation in northern China provide strong evidence that early eukaryotes were already performing oxygen-producing photosynthesis. These rocks contain molecules such as phytane and pristane, which come from chlorophyll A, the same pigment used by modern algae and plants to capture sunlight. The appearance of these compounds also lines up with a rise in phosphate and molybdenum in the oceans, two key nutrients that help drive biological growth. Together, these clues show that by this time, algae were thriving, and Earth's oxygen cycle was becoming increasingly influenced by eukaryotic life. Later, Secondary and tertiary endosymbioses multiplied photosynthetic diversity. Eukaryotes such as diatoms, dinoflagellates, and euglenids captured whole algal cells, creating nested organelles with 3 or 4 membranes. This proliferation of plastids transformed marine ecosystems and, eventually, atmospheric composition.
The architecture of complexity.
Mitochondria and plastids were only part of the story. Within eukaryotic cells, a dynamic cytoskeleton of actin and microtubules enabled internal transport, phagocytosis, and cell division. Vesicles budded from endomembranes, giving rise to the Golgi apparatus and endoplasmic reticulum. The nucleus isolated transcription. Chromosomes wrapped around histons and recombined through meiosis. The cell had become a community within itself. The development of the cytoskeleton, the internal framework that gives cells shape and movement, was a major step in the evolution of complex life. Key proteins such as actin and tubulin, which form the filaments and tubes inside modern cells, can be traced back to ancient relatives found in a group of microbes called Lokiarchaeota. When these proteins link together, they create flexible structures that allow cells to move, change shape, and even engulf food particles, a process known as phagocytosis. This ability to take in nutrients and form close partnerships with other cells set the first eukaryotes apart from simpler, rigid-walled microbes, paving the way for greater complexity. Some of the first large microscopic fossils of complex cells, including forms known as Topania, Valeria, and Arcticellularia, show intricate surface patterns that suggest they already had an internal cytoskeleton to shape and support their cell walls. These fossils, dated to between 1.63 and 1.45 billion years ago, appear at the same time as chemical evidence showing a rise in biological activity and oxygen levels in the oceans. Shifts in the carbon isotopes from this period point to more active photosynthesis and carbon cycling, indicating that Earth seas were becoming increasingly rich in life.
The eukaryotic expansion.
By 1.4 billion years ago, eukaryotic life had spread through coastal oceans. Diverse protists, amoebae, flagellates, early algae, inhabited microbial mats and planktonic communities. They fed on bacteria, recycled nutrients, and oxygenated the photic zone. Though invisible to the macroscopic world, they began to reshape the biosphere from the bottom up. Between 1.6 and 1.2 billion years ago, the rate at which organic carbon was buried in sediments rose by about 30%, according to chemical evidence from ancient rocks. This means that living organisms were becoming more effective at trapping carbon in the deep ocean and seafloor, a process sometimes called the biological pump. As more carbon dioxide was locked away, oxygen levels in the atmosphere rose slightly. This long-term feedback between life and the environment helped create the conditions that would eventually allow multicellular animals to evolve. The geologic record captures the spread of these ecosystems in laminated shales and black mudstones rich in organic matter. Biomarkers include steranes, hopanes, and isoprenoids indicating both producers and heterotrophs. These strata underpin the mid-Proterozoic sedimentary quietude, tectonically calm, biologically steady, yet slowly oxygenating.
Transition summary.
Through endosymbiosis and internal reorganization, eukaryotes solved the energy problem and invented cellular architecture. Within them, life became compartmentalized, powered, and capable of innovation. The quiet billion years that followed were not barren stagnation but the gestation of complexity itself.
Part 3. The invention of sex and early multicellularity. The invention of sex.
Complex cells brought with them an opportunity unknown to the microbial world, the ability to combine, exchange, and reshuffle genes. Somewhere between 1.6 and 1.2 billion years ago, eukaryotes evolved meiosis, the process that halves chromosome numbers and allows gametes to fuse. Through sexual reproduction, genetic information could be recombined each generation, producing endless variation for natural selection to act upon. The world's first sexual lineages appeared long before animals, flowers, or spores. Modern genetic studies show that all major groups of complex life, including animals, plants, fungi, and single-celled protists, share the same set of core genes that control sexual reproduction. These genes, which guide the process of meiosis, the division of cells to form sperm and eggs, are so similar across all eukaryotes that scientists believe sex evolved only once, in the common ancestor of all complex life. Molecular dating of these genes places this breakthrough between 1.6 and 1.4 billion years ago. This timing matches geological evidence showing higher levels of key metals like zinc and copper, which these enzymes require, and signs of increased nutrient recycling in ancient marine sediments, a sign that Earth's ecosystems were becoming more dynamic and interconnected. The key advantage of sexual reproduction is that it allows genes to be reshuffled each generation, creating new combinations that help species adapt. In contrast, organisms that reproduce asexually simply copy their DNA, which means harmful mutations can build up over time, a process known as Muller's ratchet. By mixing genetic material from two parents, sex effectively resets the genome, repairing damage and introducing useful variation. Computer models show that for small organisms like early eukaryotes, which typically lived in populations of about 100,000 to a million individuals, sexual reproduction could double the rate of beneficial genetic change, even though it required more effort and produced fewer offspring overall. The earliest clear evidence of sexual reproduction in the fossil record comes from a species called Bangiomorpha pubescens, found in rocks in Arctic Canada that are about 1.05 billion years old. These fossils belong to a type of red algae made up of thin, thread-like filaments. Within them, scientists have identified structures that look like spores and reproductive organs known as gametangia, showing that the organism alternated between two life stages, one with a single set of genes and one with a double set. This discovery marks the oldest known example of both sex and multicellularity in the same organism, representing a major milestone in life's evolution.
The road to multicellularity.
Life's next great leap was cooperation. Once cells could adhere and communicate, they could divide labor, forming structures larger than any individual. The earliest experiments in multicellularity likely occurred many times and independently, in red and green algae, fungi, and protoanimals. At first, these colonies were simple filaments or spheres, held together by extracellular polymers. Later, they developed specialized tissues. The genetic foundations for multicellularity predate its appearance. Comparative genomics reveals that unicellular choanoflagellates and vulvacine algae already possess adhesion, cadherin-like, signaling, tyrosine kinase, and differentiation, transcription factor, genes homologous to those in animals and plants. These genes were co-opted from ancestral roles in cell-cell recognition and prey capture. Fossils of multicellular life older than 1 billion years show that complex organisms appeared far earlier than once thought. One striking example is Gabonionta, found in 2.1 billion-year-old rocks in Gabon, Africa. These fossils are a few centimeters across and have lobed, organized structures that suggest the cells grew and developed together in a coordinated way. Although scientists are still unsure exactly what kind of organisms they were, these fossils prove that multicellular organization evolved long before the famous Cambrian explosion. Later examples, such as Bangiomorpha, a red alga about 1 billion years old, and Paliastrum, a green alga about 900 million years old, show that multicellularity arose more than once, especially among early photosynthetic life forms. Experiments with modern green algae, the Volvox lineage, illustrate evolutionary steps mirrored in the fossil record, from single cells, Chlamydomonas, to colonies of identical cells, Gonium, to differentiated spheroids, Volvox. Each stage adds a single genetic innovation, adhesion, polarity, cell cycle coordination, showing that complexity can arise gradually through co-option, not sudden design.
Oxygen and nutrients, catalysts of complexity.
During the middle part of the Proterozoic eon, Earth's environment placed strict limits on how life could evolve. Oxygen levels in the atmosphere and oceans stayed low for hundreds of millions of years, but every so often, around 1.4, 1.1, and 0.8 billion years ago, brief whiffs of oxygen appeared. Each of these oxygen spikes coincided with periods when complex eukaryotic life began to diversify. Scientists think these bursts of oxygen followed times of continental uplift and increased rock weathering, which released nutrients like phosphorus and iron into the oceans. These nutrients boosted biological activity, creating short-lived zones of higher oxygen that helped drive evolutionary progress. Chemical clues preserved in ancient marine rocks reveal how oxygen levels in Earth's oceans gradually increased over time. Between 1.6 and 1.0 billion years ago, the amount of the metal molybdenum in ocean sediments rose from very low levels to much higher ones, showing that deep waters were becoming periodically oxygenated. At the same time, variations in chromium isotopes point to bursts of oxygen-driven weathering on land. These chemical spikes line up with major biological milestones: the rise of red and green algae, the growing diversity of acritarch microfossils, and the first appearance of microscopic remains rich in sterols, compounds found in the cell membranes of complex life. Phosphorus, a key nutrient for all living things, played a major role in controlling how much life the oceans could support. As continents eroded and rocks rich in the mineral apatite broke down, phosphorus was released into rivers and seas, fueling the growth of algae. Scientists call this process the phosphate feedback. As more oxygen spread through soils and surface waters, more phosphorus became available, which in turn stimulated algal blooms. When these algae died and were buried, they locked away carbon and allowed even more oxygen to build up in the atmosphere. This positive feedback loop helped push Earth's environment toward greater biological complexity.
Early eukaryotic ecosystems.
By 1 billion years ago, shallow marine platforms hosted complex microbial mats composed of cyanobacteria, algae, and heterotrophic protists. Stromatolites became less common, replaced by layered microbial algal communities that stabilized sediments and formed the first benthic ecosystems. Planktonic algae filled the photic zone, driving new carbon and nutrient cycles. After about 1.1 billion years ago, the chemical makeup of carbon in ancient rocks began to vary much more than before, showing that life was becoming more active and that more organic material was being buried in sediments. Black shales from this time contain chemical fossils called steranes, which come from red and green algae, clear evidence that photosynthetic eukaryotes were becoming more diverse. At the same time, changes in the ratios of nitrogen isotopes reveal that Earth had developed a fully functioning nitrogen cycle, with processes like nitrification and denitrification that depend on oxygen. Together, these clues show that by this point, both the biosphere and atmosphere had grown far more dynamic and oxygen-rich than ever before. Microscopic predation emerged. Fossils of Melanocerillium and Paleomastus show openings interpreted as feeding structures. The evolution of phagocytosis among protists fundamentally changed nutrient flow, recycling organic matter and linking food webs for the first time. Ecology had begun in earnest.
The emergence of differentiation.
Once oxygen, energy, and genetic diversity converged, some lineages experimented with cellular specialization. Colonial organisms divided labor between somatic and reproductive cells, paving the way for tissues. Communication evolved through chemical signaling, precursors to hormones and neurotransmitters. The cell had learned cooperation. Groups of genes that control how cells develop and specialize, known as homobox and MADS-box genes, first appeared around 1 billion years ago. These genes act like master switches, turning other genes on and off to guide the formation of different cell types and structures. The earliest evidence of their use comes from red algae, where they help regulate the production of spores and gametes for reproduction. These genetic innovations arose through a mix of gene duplication and horizontal gene transfer, the sharing of DNA between different organisms. Once sexual reproduction evolved, these new genes could spread and combine in new ways, driving the emergence of greater complexity in early eukaryotic life. Depth cutaway. In rocks from the Torridonian group in Scotland, dating to about 950 million years ago, scientists have found fossils of multicellular algae up to 300 micrometers wide. These tiny organisms show clear signs of cell walls and organized patterns of cell division, evidence that their growth was carefully coordinated rather than random. Preserved in sediments from ancient river deltas and lakes, these fossils record a key evolutionary moment, the shift from simple microbial mats to the earliest forms of tissue, representing life's first steps toward organized, multicellular structure.
Transition summary.
By the close of the Mesoproterozoic, life had reinvented itself once again. Sex introduced shuffling and innovation. Multicellularity introduced structure and cooperation. Oceans once dominated by single cells now contained colonies and filaments, photosynthetic forests of microscopic complexity. The stage was set for the next transformation, the Ediacaran world, when multicellular life would fill the seas with form.
Part 4. The Ediacaran Dawn and Prelude to Complex Life. The Ediacaran Threshold.
By 900 million years ago, Earth's biosphere was ready for another leap. The long stability of the Mesoproterozoic began to break. Supercontinents drifted, climates fluctuated, and oxygen levels crept upward. In this changing world, the quiet lineage of eukaryotes, once confined to microscopic realms, began to expand into visible forms. Sheets, filaments, and disks of soft-bodied organisms colonized the seafloor. Their traces mark the dawn of macroscopic life, the Ediacaran period, bridging the gap between the microbial Earth and the animal world to come. The Ediacaran period, which lasted from about 635 to 541 million years ago, began after the dramatic global ice ages of the Cryogenian, when much of Earth had been frozen over. As the planet thawed, oxygen levels in the atmosphere rose sharply, possibly reaching up to 1/5 of what we have today. At the same time, melting glacier released nutrient-rich runoff from the continents into the oceans, fueling an explosion of biological activity. Chemical evidence from carbon isotopes shows that organic matter was being produced and buried at high rates, meaning the carbon cycle had become much more dynamic. Together, these changes created the oxygen-rich, fertile conditions that allowed complex multicellular life to flourish for the first time.
Continental motions and climate instability.
During the Neoproterozoic era, the giant supercontinent Rodinia slowly came together and then broke apart again, reshaping Earth's surface. As the continents drifted, new coastlines and shallow seas formed, changing ocean currents and climate patterns. The breakup also exposed vast areas of rock to weathering, which pulled carbon dioxide out of the atmosphere and may have caused two extreme global ice ages: the Sturtian, around 720 million years ago, and the Marinoan, around 635 million years ago. These snowball Earth events nearly froze the planet solid, posing huge challenges for life. Yet when the ice finally melted, the newly exposed rocks released nutrients into the oceans, sparking a surge in biological growth that helped set the stage for the rise of complex life. Chemical clues preserved in ancient rocks reveal how dramatically Earth's environment shifted during and after the Great Ice Ages. Layers of carbonate rock that formed directly above glacial deposits show very low carbon isotope values, a sign that huge amounts of buried organic material were rapidly oxidized once the planet began to warm. At the same time, changes in strontium isotopes point to a period of intense erosion and weathering on the continents as the ice melted. These chemical signals coincide with a surge in sterane molecules, which come from algae, showing that photosynthetic life quickly rebounded in the newly fertile oceans after each global thaw. Computer models of Earth's ancient climate show that it took an enormous buildup of carbon dioxide in the atmosphere, more than 300 times higher than today's pre-industrial levels, to melt the planet out of its global ice age. When the ice finally gave way, this intense greenhouse effect caused surface temperatures to soar above 40 degrees Celsius for thousands of years. The sudden heat put great stress on surviving ecosystems, but it also drove a burst of chemical activity and rapid environmental change, creating new opportunities for evolution and the emergence of more complex life.
The second oxygenation.
After the planet thawed from the Marinoan Ice Age about 635 million years ago, Earth's environment underwent another major transformation known as the Neoproterozoic oxygenation event. During this time, oxygen levels rose higher than ever before, finally reaching amounts that could support large, energy-demanding organisms. This surge in oxygen changed the chemistry of the oceans, making them more hospitable to complex life. As a result, the first multicellular animals began to appear, along with the earliest interconnected food webs, marking the beginning of a new and dynamic stage in Earth's biological history. Chemical evidence from ancient rocks shows that by about 580 to 560 million years ago, Earth's deep oceans had finally become rich in oxygen. Measurements of iron and molybdenum isotopes in dark shale layers reveal that oxygen had spread throughout the seafloor, while chromium isotopes indicate that oxygen-driven weathering was also happening widely on land. Around the same time, large phosphate-rich deposits, such as those found in the Dushantuo Formation in China, began to form, showing that both oxygen levels and biological productivity had increased. The oceans also became richer in sulfate, reaching about 10% of modern levels, which supported new types of sulfur-based metabolisms and helped early organisms begin to build shells and skeletons. This rise in oxygen was closely linked to changes in life itself. As large photosynthetic algae became more common, they absorbed carbon dioxide and buried more organic carbon in ocean sediments, which allowed oxygen levels to rise. Higher oxygen, in turn, made it possible for larger organisms to evolve, ones that needed more energy and oxygen to survive. This created a powerful feedback loop. More life produced more oxygen, and more oxygen allowed life to become more energetic and complex, setting off one of the most important evolutionary chain reactions in Earth's history.
Eukaryotic diversification.
With both oxygen and energy now plentiful, eukaryotic life began to diversify at an astonishing pace. Red, green, and brown algae spread across the oceans, forming vast underwater forests, while fungus-like organisms began to grow within sediments on the seafloor. For the first time, predatory single-celled organisms appeared, hunting and consuming other microbes. Fossils from between 1 billion and 600 million years ago show an incredible variety of acritarchs, tiny, intricately patterned microfossils that represent the plankton of that era. Their growing size, complexity, and global abundance mark a major ecological turning point, the worldwide expansion of complex eukaryotic life. The variety of acritarchs, tiny fossilized cells thought to represent ancient plankton, expanded dramatically between 900 and 700 million years ago. Studies of hundreds of rock formations around the world show that what was once a time of low diversity, with fewer than a dozen types in each sample, gave way to a period bursting with over 100 distinct forms. Detailed measurements reveal that some of these cells grew up to 0.5 millimeter across and developed thick, multilayered walls, features that would have required far more energy and oxygen to sustain. This explosion in size and complexity reflects how rapidly life was responding to the newly enriched, oxygenated oceans of the Neo-Proterozoic world. Chemical traces known as biomarkers provide a hidden record of life that existed long before animals left visible fossils. Compounds such as 24-isopropylcholestane, made by sponges, and dinosterane, produced by dinoflagellates, a type of advanced plankton, have been found in rocks dating from 635 to 600 million years ago. These molecules act as molecular fossils, revealing that early animals and complex single-celled organisms were already living in the oceans well before they appeared in the fossil record. They mark the dawn of visible life, the first hints of the larger creatures soon to come.
Experiments inform the Ediacaran organisms.
By about 575 million years ago, the fossil record reveals the first communities of large, soft-bodied organisms, the Ediacaran biota. Found in the sandstones and siltstones of Newfoundland, Namibia, and Australia, these organisms show quilted, frond-like, or radial patterns unlike any modern species. Some resembled ferns anchored to the seafloor, Charnia, fractifusus, others with discs or pouches embedded in microbial mats. They may have absorbed nutrients directly through their surfaces, lacking mouths, guts, or skeletons. Some of the most beautifully preserved fossils from the Ediacaran period come from the Mistaken Point Formation in Newfoundland, dating to about 565 million years ago, and the Ediacara Hills in South Australia, about 555 million years old. These sites are precisely dated using uranium lead measurements from volcanic ash layers found between the fossil beds. The fossil surfaces reveal entire communities of organisms, with individuals aligned in similar directions and showing a range of sizes. patterns that suggest these early life forms interacted with one another and may even have reproduced sexually. Chemical evidence from the rocks where these fossils are found shows that the oceans at the time were rich in oxygen. Measurements of iron and molybdenum confirmed that even the deepest waters had become fully oxygenated. A large shift in carbon isotope values, known as the Shuram anomaly, suggests that huge amounts of buried organic matter were suddenly oxidized, which briefly lowered oxygen levels but also released nutrients into the oceans. These environmental ups and downs may have acted as a spark for evolution, driving the rapid diversification of multicellular life that defines the late Ediacaran world.
The Ediacaran ecosystem.
Ediacaran seafloors were microbial landscapes. Thick mats of cyanobacteria and algae-bound sediments, forming stable surfaces on which larger organisms could attach. Within these mats, chemical gradients supported diverse microbial communities, sulfur reducers below, oxygenic phototrophs above. The new macroscopic organisms exploited this layered ecosystem, grazing, absorbing, or symbiotically associating with microbes. Trace fossils, grooves, burrows, and feeding trails first appear in the late Ediacaran. Simple and branched tubes, planolites, triptychs, mark the earliest evidence of locomotion and behavior. They record oxygenated substrates supporting active metabolism. These tracks prefigure the rise of bilaterian animals in the Cambrian. Chemical fossils preserved in Ediacaran age rocks show that by this time, Earth's ecosystems had become both balanced and complex. Molecules called steranes, from eukaryotes, and hopanes, from bacteria, appear in roughly equal amounts, suggesting that both kinds of life were thriving together in stable, energy-rich environments. At the same time, the ratios of nitrogen isotopes indicate that the planet had developed a fully functioning nitrogen cycle, where oxygen played a key role in recycling nutrients. This system could support multiple levels of the food chain, marking an important step toward the modern, interconnected ecosystems we know today.
The genetic foundations of animal life.
Genetic studies show that the blueprint for building animals existed long before the famous Cambrian explosion. Many of the key genes that control how cells stick together, communicate, and specialize, such as integrins, cadheins, and the WNT, notch, and hedgehog pathways, first evolved in single-celled organisms related to animals, including choanoflagellates and philisterians. These early microbes used the same genes for simpler purposes, like attaching to surfaces or sensing their surroundings. Later, evolution repurposed this ancient genetic toolkit to create tissues, organs, and body plans, laying the groundwork for the first multicellular animals. Genetic comparisons suggest that animals split from their single-celled ancestors sometime between 800 and 750 million years ago. Chemical fossils called 24-isopropyl cholestanes, produced by early sponges, appear in rocks from the same period, supporting this timeline. By then, oxygen levels had reached about 10 to 20% of modern concentrations, enough to fuel the energy needs of multicellular organisms. The fossils of the Ediacaran period, though often mysterious in form, capture the first flourishing of these new kinds of life, creatures made possible by both rising oxygen and an increasingly sophisticated genetic toolkit. Experiments on modern sponges, such as amphimedon, show that even with a relatively small set of about 18,000 genes, they can still form distinct tissues and regulate a simple immune system. This suggests that the basic genetic machinery needed to build and maintain a multicellular body is surprisingly minimal. These ancestral genetic systems probably first appeared in Ediacaran age organisms, representing an important evolutionary bridge between simple microbial colonies and the first true animals.
The end of the Ediacaran.
Around 541 million years ago, the Ediacaran world vanished. Changing ocean chemistry, rising predation, and ecosystem restructuring erased its softly bodied organisms. Yet their legacy endured. They had proven that complex multicellular life was possible. Within a few million years, the Cambrian explosion would fill the seas with shells, skeletons, and motion, the visible world of animals. Around 541 million years ago, Earth underwent a major environmental and biological transformation. many of the distinctive Ediacaran organisms disappeared, while new trace fossils show that animals capable of burrowing and moving through sediments had emerged. At the same time, the chemistry of the oceans changed dramatically. Sulfate levels doubled, and rising calcium carbonate made it easier for creatures to develop shells and skeletons. Shifts in carbon and sulfur isotopes reveal that the planet's carbon-sulfur cycle was being reorganized, likely due to expanding zones in the ocean with little oxygen. Although these changes disrupted existing ecosystems, they also set the stage for the rise of energetic, complex animals that would soon dominate the Cambrian world.
Conclusion: the hidden revolution.
The Proterozoic eon, long dismissed as uneventful, was in truth the crucible of complexity. In its billion years of quiet, life discovered partnership, sex, and cooperation. Cells merged to form organelles, individuals joined to form tissues, and genes evolved into networks capable of building bodies. By the close of the Ediacaran, the blueprints of every later organism already existed in microscopic form. The next chapter, the Cambrian explosion, would make this hidden revolution visible.